In our everyday lives, we are interacting with millions of organisms that we can't even see. There are multiple kinds, but I'm mainly going to talk about bacteria. Recently, after the COVID-19 pandemic, much of the focus went to viruses. But there are so many bacteria which are useful and bad for us, and they live in all sorts of conditions. Some of them live where we stay in normal temperatures, some of them live in extreme temperatures, some of them live in other kinds of extreme conditions, extremely salty conditions. Thermophiles live in extremely hot, like geysers and those type of places, which is actually important because we isolated PCR. For PCR, we need a polymerase called Taq polymerase. For COVID-19 testing, this was taken from one of these, which is quite important. But do you think viruses infect only human beings? Bacteria have been at war with viruses. Viruses and bacteria have been at war since the dawn of time. They attack each other. Viruses don't just attack humans, they also attack bacteria. The viruses that attack bacteria are called bacteriophages. The word phage means devour. The word literally quite translates to organisms that devour bacteria. What is a bacteriophage? Bacteriophage is a type of virus. It has only two components. It's very simple, unlike our cells. Very simple. It has something called genetic material, which is encapsulated in a protein caspid. So its genetic material can be of two kinds. It can either be RNA or DNA. This is the basic structure of a bacteriophage. This is different parts of the skeletal body of it, and inside, in the head, is DNA. It's genetic material, or RNA actually. And this outside everything is called the protein caspid, which protects the DNA and RNA. Why do the DNA and RNA need to be protected? They're very sensitive to temperature and pH changes, so they need to be safeguarded that way. Now, why do bacteriophages infect bacteria? What do they gain from it? Nature simply just doesn't do things. There is a reason for bacteriophages to infect bacteria. Okay, let's go back to one thing. Viruses, are they living or non-living? Here are some characteristics that say that are contradictory in nature actually. You could say that a bacteriophage or a virus is a living thing because it has genetic material. It can mutate. We've seen that happen, like we have multiple waves of COVID-19, mutated versions, that's possible. They can also multiply, which means reproduce. These are all the characteristics of living things. How about this? Can living beings, can we humans be crystallized and stored in a freezer for 10 years and be brought out back to life? That doesn't happen with us. But with these creatures, you can do that. With these organisms, they can be stored for a really long time. And they don't interact with us if they're outside of us. Do we see a bacterium coming and saying stuff to us or coming and irritating us during our day? That doesn't happen. So what are they? They're actually considered the link between living and non-living. They can be both. You may ask how. Outside of our body, outside of a host, they are dead. Essentially, they're non-living. They cannot do anything. They're inert. But inside of a host, they become alive. Why do they need the host? Why do they go to the host? What is the purpose of any creature if you talk from a biological perspective? It is to reproduce, multiply, and propagate its DNA around. For that purpose, bacteriophages infect the host cell in order to multiply. How does this happen? Viral multiplication. This is very simple. This is my fruity bacteriophage sitting on top of a virus bacterial cell. This is a bacterial cell and this is the bacteriophage. It goes and sits on the bacterial cell and then it inserts. It has an injector kind of thing around here. It inserts this genetic material. It travels down and goes out. It goes out it releases into the bacterial cell. Now the bacterial cell also has some type of genetic material already in it, which is a blue thing in my diagram. The next step, this DNA, the bacteriophage or the viral DNA multiplies inside of the bacteria. It uses all the resources of the bacteria to multiply. Like I said, these are very, very simple organisms. They do not have much of anything. Our cells, the bacterial cells, are more complex in nature, and they have more resources. That's the word, resources. It uses all the information and the mechanisms of a bacterial cell to propagate itself. What's happening here is a new phase. Parts of the new phage are made, and they begin their assembly. Now, once they assemble inside, like this is they look very similar, right? The cycle is completed. Once it's completed, biomaterial inside the bacteria wants to travel. So it breaks open. It goes out of the cell and it travels. This is called host cell lysis. Lysis is breaking of something. If the cells of a host break, what does that mean? They're dying. They will die. They will not survive. A bacteria is a single cell organism, so it will die. Inside our cell, some of our cells will die and the infection will spread within our system. That's what will happen. In viral multiplication, there are two types: lytic cycle and lysogenic cycle. Lytic cycle is basically what I've described. Step by step, this is what happens. In lysogenic cycle, they come and they multiply inside the bacteria, but they pause and they wait. They wait for the environment to become suitable for them, and then they continue the next steps and release the same. Most of the bacteria cannot survive a viral or a bacteriophage attack because even they aren't that advanced. They do not have such great protective barriers. We have so many barriers and we still fight all sorts of infections. But this is where things get interesting. What if a bacterium survives? What happens then? Right now I said weak, right? A bacteria cell is weak. But no, it has a very interesting and strong immune response. If it survives the initial attack, then we are not the only organisms with an immune system. Even others have developed immune systems. A bacteria has an adaptive immune response called Crispr. Crispr, C-R-I-S-P-R. I'll get into the details of that in a while. What it does is basically, it stores the DNA within it to get back the next time of it. So, yeah. So CRISPR is short for, CRISPR is the acronym for Clustered Regularly Interspaced Short Palindromic Repeats. Huge word, I know, but they make sense. If a bacterium survives this attack, imagine the green DNA to be the bacterial genetic material and the yellow one to be the viral, the phage genetic material. What it does is it integrates it into itself. It makes it a part of its own genome, in its own blue circle. It keeps it. It stores this at a site called CRISPR site. Now, this is double-stranded, two-stranded DNA. What it does is it makes an RNA form of the same thing, single-stranded RNA. The highlight of this mechanism, the highlight of the CRISPR mechanism is the Cas9 protein. It's a protein which is very crucial. CRISPR basically acts as a genetic vaccination card. It acts as an archive of viral DNA. Imagine you take a picture of every criminal and keep it with you, so that the next time if you encounter them, you know and you can identify them. Now, if the same bacterium is infected with the same bacteriophage again, this entire immune response kicks in. What happens? Viral attack, the bacteriophage comes and sits on top and injects its genetic material into it. Now, it doesn't exactly go like it did before. This entire system, this immune response is activated. This is called the RNA Cas9 Sentinel complex. Tough words, they don't mean that much. It's basically that what this Cas9, think of Cas9 and RNA as police and one car. Police and the picture. You have the picture, which is the RNA, and the Cas9, which is the police. Now this police goes around the DNA of the bacteria to identify, is this DNA stored stored anywhere. Can I identify what was injected right now? Like a new, one second, yeah. The new genetic material got penetrated, right? Now this Cas9 is trying to look, is there anything stored? Is there anything similar to the picture that I have in my hand? Where can I find it? And when it does find it, it's hunting for a matching sequence. What it does is, this is like a two-stranded thing, right? Imagine this, if I attached it here, if this was the attachment, what happens? It makes a double-stranded break, a break here and a break here. It makes a double-stranded break in the DNA helix, which activates and tells, there is a virus that's infecting us, come on, let's kill it. This basically kills the virus. You're going to the virus and breaking its DNA. Once you break the very material in the core of it, it cannot survive, right? So the virus is defeated. But this doesn't stop there. How is something that happens in a microscopic level impact us? It impacts us because this system, this immune response is programmable. This immune response can be extracted from them and used in all sorts of scenarios. It makes genome engineering a reality and not science fiction anymore, which is kind of amazing if you think about it. Something that's happening at a microscopic level can change the entire being of us. A small difference can change the entire thing. How does this come to being with, if you think in a human sense, how does this happen? Basically, cells can detect broken DNA. If any DNA is broken in our body, they detect it and they repair it. They repair it in two methods. They use two ways. They either attach the two broken ends together, simple, or they incorporate a new segment and then attach the two sides. They basically use this as a basis for programming. Imagine you could introduce a break in the DNA sequence where the faulty gene or the gene for cystic fibrosis, a medicine with no cure as of now, or a gene that causes cancer. Imagine you break that gene and that gene basically dies off. It goes away from our genetic code. What does that mean? In theory, any disease can be cured if you can identify the genetic sequence of it and snip it. We snip it and this is so precise, we can snip exactly where we want to. And then what happens is, like I said before, the cells can detect the broken DNA and this will trigger the cells to repair itself. We're giving very little stimulation to our system to make a huge impact. This technology was discovered by two people, Jennifer Doudna and Emmanuel Charpentier, who won the Nobel Prize for Chemistry in 2020. One might think genome engineering is not a new concept. Humans have been editing the gene all the way from the 1960s. We used to burst plants with radioactivity, hoping something new would come up, which is useful to us, and it did. And in the 1970s, 80s and 90s, we had so many GMOs like flavor saver tomato, BT crops. This is happening. Why is this technology deserving of a Nobel Prize? Nobel Prize is no small thing. The reason for this being, it's previously genome engineering took a long time. A lot of expertise one had to have in the field. The procedures and everything was super complex. The experiments to run everything. Most importantly, it was very, very, very expensive. What this does is any basic lab, you can even do this at home with a kit, which is quite inexpensive if you think of its implications. It's cheap, it's easy to learn, and it's twice as fast. In the future, it could be possible that you go to the hospital with a disease, with a disorder, and 2 injections is all you need. How cool would that be? How cool would that be if you went to the hospital and said you had cancer and got an injection, and that's the end of the story. That's very interesting, right? That's like pretty damn cool. But like everything, this has two sides. It has two stories. When one thinks of engineering a human being, first thing that comes to people's mind is not, oh, let's cure cancer or let's cure cystic fibrosis. Most people don't even know what that is. People will think of how do I become 6 feet 7 inches, or have blue eyes, or make sure that I'm thin, or IQ, high IQ, get into any school that I want to. Parents thinking like this for their children. This is also a possibility. This possibility of superhumans is there. There is so many ethical questions regarding this entire phase. And that's what these two scientists responsibly asked once they won the Nobel Prize. That's what they did with their new attention that they were getting. That's what they did with it. Just because science allows us to do something, should we do it? Thank you.